A review of protein synthesis. So these are 4 handouts along with a puzzle that we worked with in class, and I want to give you a teacher review in case you need to review it on your own or are not present in class when we do this. So in step number 1, they want us to get used to the pieces of the puzzle and what they're called and how it relates to everything we've learned so far. So under A, we're going to label that a base, sometimes referred to as a nitrogen base in some of your materials. You also hear us refer to them as nucleic acids. So we've got the first line, base. The second line, the C, is a base, knowing that they also could have chosen to put a T or a G or a U up there and also called those bases. And then the blue piece, this piece off to the right with no lettering in it, is a sugar phosphate unit. We talk about how that is the part of the molecule DNA, the deoxyribose part. We go into number 2, and they want to check our understanding of the pairing rules. You were introduced to this with the Amoeba sisters, apple in a tree, car in a garage. So on the left, A pairs with T, C with G, T with A, and G with C. And you can verify that by seeing that those are the puzzle pieces that fit together. So on the right side of the diagram, they give you half of DNA, A, G, C, T, G, T, just reading down the left side. And on the right side, you are to fill in the matching puzzle pieces. And going down the right, you would have a T with the A, and then a C, and a G, and an A, and a C, and an A. And again, A with T, G with C. Okay. Moving on to the replicating DNA side, what we're doing is we've unzipped the DNA molecule. It was two-sided. We opened it up to be 2 separate single-sided pieces. And your job is to fill the inside back up with the matching puzzle pieces. So as you can see in the diagram, on the left, A with T, G with C, C with G, T with A, G with C, T with A. And on the right, also A with T, G with C, C with G, T with A, G with C, T with A. So what has been created, what they want you to notice is that we now have 2 exact copies. The left and the right are identical. Is DNA always 6 base pairs long? No. If this were to make a human, we would need about 4 billion base pairs to do that, from what I've been told. Where in the cell does this occur? DNA is in the nucleus of eukaryotic cells. What is this process called? DNA replication. For what reasons would the DNA of a cell need to be copied? Well, if you think back just to you used to be a single cell, and now you're trillions. So how is it that one cell can make trillions and every one of those cells know what to do? That is because the DNA must copy itself for every one of these new cells. We could also add in things like cell repair, when things need to be replaced. Okay, modeling a message. Now DNA has another job to do, and that is to control activities in the cell. So instead of just making copies for new cells, it now needs to be able to send messages to control activities. And here, the DNA unzips again, but instead of making exact copies on the opposite side, RNA, in this case a messenger RNA, the little M in front of RNA indicates messenger, is going to dock up next to the DNA and take a message out to the ribosome. And we learned about ribosomes back in October with cell organelles. So in this case, the pairing rules are almost the same, with the exception that T is not present in RNA, but instead uracil or a U. So we can never have a T in a strand of RNA. We replace it with U's. So in the top row, CATTA is going to get docked up by G, U, A, U, and I'm just going to keep going. A, G, 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 C, G, G, U, A, G. So C with G and A with T, but it's now a U, so A going with U. And your job is to fill in all 3 of those lines. If you need to pause the video to look at check your answers, go ahead and do that. But it's the same process. Number 2, we didn't spend much time on in class. We've been here before, but where is the messenger RNA formed? Well, it goes into the nucleus, meets the DNA, and where protein is made is out in the cytosol or cytoplasm of the cell where the ribosomes are, outside the nucleus. So I just kind of color-coded those in, or well, used a pattern since I don't have colored pencils. Code makes a product. this is completed on the second day, and what we're seeing here is every 3 letters of the RNA messenger cause the tRNA, which we've seen in the Amoeba Sisters video, to bring over an amino acid and attach it to make a protein. So what we do is we use this chart, and we have to find the 3-letter sequences between the dashed lines. So in 1A, they give you a GUA, and you have to find GUA in the table on the right, in the mRNA codons, we call them, the 3-letter code. And in this case, it's valine, and valine has the abbreviation VAL. The next 3 letters, CUA, leucine, and the abbreviation they have is LAU, and then GGG is glycine, GLY, and GCU is alanine, ALA, and then UAG codes for stop. In other words, it tells the RNA, or tells the ribosome, that this is the end of the protein chain, stop making it, cut it loose, and send it off where it's supposed to go. So your job is to finish B and C. Again, the answer key's there. But all we're doing is taking the existing 3 letters from the messenger RNA, finding it in the codon chart, and writing down what amino acid it is in abbreviated form instead of the whole word. And number 2, you're working backwards. Now, as you can see in the codon chart, there's more than one 3-letter codon for each amino acid. So you can choose. So even though I have an answer key up here, basically I got lazy and picked the first 3 letters every chance I had, but you could pick any of the letter combinations listed, so there could be too many correct answers to cover in this review video. But so phenylalanine, which is PHE, is in the chart, and the first 3-letter codon for it is UUC. So I write down UUC, the first 3 letters of the messenger RNA that would take it, and then I go on to valine, and I pick GUA because it was the first in the list, but again, you could pick any of those. And then I continue on to fill up the whole chain. Now part B is also going to have too many answers to review, but it does need to be based on your answer in A. So since I have A written UUC, GUA, there is a single correct answer to match my answer in A, and that is by pairing A with T, G with C, or A with U, G with C, you get your original DNA strand. So since I have UUC, well, U goes with A, U goes with A, G goes with C, so my first 3 letters of DNA would be AAG, and then CAT, and then TTG, GTT, on and on until I've matched the entire message strand. And then number 3, that's just kind of an extension, so answers of your choice there. There's nothing to cover in terms of right or wrong, just making sure give you time to be confident in what you know. So that covers the structure of DNA, how DNA replicates itself, how DNA makes proteins, and proteins are and do all the things in our bodies, and that is how we make this leap from DNA controlling cells.